PhenoStacks is a visual analysis tool that helps genetics researchers and specialists explore the phenotypes of rare disease patients in cross-sectional cohorts. PhenoStacks is designed to facilitate analysis through a rich, interactive, multi-view interface. Phenotypes are observable and measurable patient traits that describe all the symptoms and features of a rare genetic disease. They provide rich insight into the multitude of factors affecting genetic diseases. Formative interviews with genetics researchers were used to elicit common cross-sectional cohort analysis tasks. Using Bremer and Munzner's task typology, we identified a set of abstract visualization tasks to support. Explore, summarize. The Details panel displays phenotype observations across patients in an observations plot. This matrix-like visualization shows phenotypes in rows and patients in columns. Phenotypes observed as being present are denoted in purple, and those observed as being absent in blue. Phenotypes without recorded observations are in gray. The observations plot can be sorted by phenotype and patient attributes. For example, sorting by co-occurrence clusters similar phenotypes and patients closer together. Locate, identify. The Summary panel displays metrics of phenotype observations within a cohort and between cohorts, using space-filling radial hierarchy charts. This enables geneticists to answer questions such as, what are the most common phenotypes in this cohort? Or, which phenotypes are different between these two cohorts? Browse, compare. By leveraging the topology of the Human Phenotype Ontology, phenotypes can be hierarchically grouped by semantic similarity. Due to multiple inheritance of the ontology, phenotypes falling within more than one categorization are duplicated. To eliminate these duplications, we introduce an ontology simplification algorithm that selects the most diagnostically significant categorization for each phenotype. Feedback indicated that our novel simplification algorithm produced categorizations that were preferred. We evaluated PhenoStacks during a deployment study with 4 researchers using their own data. The researchers found that visual analysis of phenotype patterns was applicable to a broad range of tasks. By sorting patients and phenotypes, PhenoStacks could help identify phenotype patterns to differentiate new disease subtypes or identify markers correlated to disease severity. By exploring the observations plot, PhenoStacks could be used to assess the completeness of existing datasets or to audit the consistency of data collection. Presenting phenotypes in the context of the Human Phenotype Ontology could facilitate the analysis of disease localization and aid communication between researchers with varying specializations. PhenoStacks is available as an open source project at phenostacks.org.